Hello, thanks for attending this session. I am Rupam Bhattacharya. I am a student in the Department of Biostatistics at University of Michigan, and I will be talking about my work in precision medicine in cancer. To give you a brief background of the problem that we are looking at, precision medicine is the way where we optimize treatment for each individual patient. At a cancer context, this basically means that we will be looking at their cellular features in terms of the disease, and accordingly we will be optimizing the treatments. This is an easier problem and has some simpler solutions if you have the right kind of data. For example, if you have a data like this, where for a set of patients you have multiomics data plus some clinical information, which importantly includes also their responses to the set of drugs that you were trying to look at, then you could basically fit a training model based on that for the drug responses on whatever other omics and non-omics covariates you have, and based on that training model for a future set of patients with that same set of omics and other information, you could predict their responses to different sets of drugs, and based on those predicted responses, you could optimize what drug to use for treating which patient. In this talk, we'll be talking about proteomics, but our methods are extendable to other kinds of omics as well. The challenge really comes in here for the drug responses part. You cannot really have a lot of drugs applied to patients at your will, which has of course both ethical and clinical issues, and the maximum that you will get in such a setting is possibly only a set of a few drugs which were actually applied to a patient for the treatments. So for a group of a few hundreds of drugs at a large scale, getting responses across a decent number of patients is not really something that will be available in general. We suggest a workaround using model systems to solve this problem and to also fulfill our overall precision medicine target. Model systems are basically settings like cell lines or other kind of structures which you can have controlled in a lab environment and apply different kind of drugs or compounds and see their responses. You can also get omics and other kind of cellular data for such systems. There has been in recent years and decades a huge increase in terms of these kinds of data that we are talking about. For example, there are a lot of databases that provide you with the patient omics data. You can also have a lot of cell lines data, including their responses to drugs. I have not included the full names of these databases I have reported here, but we'll be talking more about the specific ones that we'll be using in this project. Our goals are primarily twofold. We want to bridge anti-cancer pharmacological data to the large-scale omics that we have. And while doing that, we want to utilize the patient individuality or heterogeneity in terms of our decision making. The features of this problem are, first of all, we are looking at a large set of genes and proteins which are potentially interacting between themselves in functional pathways, and they have different kinds of small and cascading effects that are possibly accumulating together to express something bigger. That has to be taken care of, and also we need to take care of the variation between and possibly within cancer lineages depending on which tissue or which organ it affects. There are also possible variations across model systems because for a cell line which is controlled in a lab, the microenvironment is going to be hugely different from a tumor which is inside a patient's body. We propose our method which is called TransPRECISE. We look at the data structured like this, where this is the part that's missing for us and we want to fill that up. We break the problem up into two different steps. The first step basically looks at only the proteomics part of the data across the patients and cell lines, and we try to compare this, find some sort of similarity so that we can identify what we call avatar cell lines for patients from the pool of all available cell lines. The second step is basically looking at just the cell lines by themselves and fitting training models for the drug responses on their proteomics information. Now, we combine these two steps to predict patient drug responses in the sense that for a given set of patients, you would first get a set of cell lines that match with them very well. On those cell lines, you will train a model, and then using that trained model, you will predict the patient drug responses from the set of patients that you initially started with. This is our overall framework from our published paper. The few important things to notice here are how we are breaking up this problem into multiple different steps. We are going into a pathway level in terms of the proteomics across all samples, that is patients and cell lines, and then fitting cancer specific networks. Then we are deconvolving that into a sample specific level, getting some summary of pathway network activities, and based on those summary scores, we are then basically identifying the avatar cell lines, and from that we are going into the drug sensitivity prediction part. This slide also had some information about the sample sizes and the cancer types that we are using, but we will be talking more about those at a later stage. So first, let me go into a bit of notations before I describe to you the mathematics of what we are exactly doing here. So we are looking at one cancer type across either patients or cell lines and one pathway combination. For that combination that we have chosen, yi denotes the n cross 1 expression vector where n is our sample size from that cancer type or site of the ith protein in that pathway. Throughout this talk, we will be using k to indicate the other proteins in that same pathway as protein I, and we will be using j across 1 to n to indicate our samples. Ci is the number of nodes that are connected to that protein I within that pathway in our fitted network, and P will be the number of total proteins in that pathway. G will be a tuning parameter which we will be using in a Bayesian variable selection setting, and we are using G equal to the sample size n. For the cancer-specific networks, which are our first stage, we use a Bayesian graphical regression model, where basically for each protein, we define a model like this, where the ith protein is regressed on all the other proteins that are available there. And the beta parameters that you see, which are basically the coefficients corresponding to the other proteins, are directed towards this protein. That conditional on the tuning parameter that we were just talking about has a normal prior, and that normal prior basically uses the Zi transpose Zi term to incorporate the information from the covariates into the prior. The sense is that if the Zi transpose Zi is really large, then this variance is really small, and then this beta I is basically concentrated almost at 0, in the sense that Zi does not really have a lot of information to specifically estimate this beta or say that it has some effect coming out of the other proteins. This prior, which is called the Zelnar's G prior, also does a bit of variable selection here. The G equal to n basically ensures that this is an unit information prior. For sample specific course, what we do is once we have fitted this model, which we do by writing down all these conditional distributions and then doing an MCMC using Bayesian model selection, we get the estimates of beta hat and sigma hat. I have not incorporated the information regarding the MCMC here, but basically you can write down the MCMC just using these conditionals, and you don't need the full marginal to write that, but you can still get estimates for that full marginal, which are denoted by beta hat and sigma hat here. We have used a package in R called BMS, which incorporates the Bayesian model averaging or BMA method for us. Once we get those beta hats and sigma hats, we can go to the sample specific course step, where we basically say that if the observed expression for the jth sample corresponding to the ith protein is within a certain tuned range around 0, then we call the probability of that conditional on the posterior as Pij0. Similar, if it's on the left side of that interval, we call it Pij negative, and if it's on the right side, we call this Pij positive. In some sense, this actually corresponds to that protein being neutral or suppressed or activated respectively for that specific patient or cell line sample that we are looking at. Once we have done that, this is at a sample cross protein level, and that we want to take to a pathway level. For that, what we do is for each sample J, we look at all the protein scores that we have computed in the previous step for one pathway, and we aggregate them in a weighted average structure like this, where the CIs basically decide that if a protein is really a hub protein and is connected to a lot of other proteins, then that has a higher say in the score. And similarly, at the other end, the proteins that are not connected with too many proteins, they have actually a lower effect on the overall weighted average that we are looking at. So once we have done this, then basically we have a sample by pathway version of these scores, and similarly we have 3 versions of them which basically correspond to that pathway being activated, suppressed, or neutral, which are denoted by the plus, minus, and 0 signs here. Once we have got this, now we can use them to aggregate and identify different kind of patterns in the omics of our samples. For example, we can add the suppressed and activated scores, which we call the network aberration scores. Based on that, comparing the network activities or just looking at simple correlation or doing a hierarchical clustering, we can say which cell lines are similar to which patients. We can also use in drug response models in the sense that for cell lines, now we can fit models using response or sensitivity to a drug as our response variables, and these pathway scores as our covariates. The specific model that we use is Bayesian additive regression trees or BARTs, and that is implemented using this R package called BART machine, which is already out there. The primary reason behind selecting this very model is that BART is good both with selection and prediction. Now we talk a little bit about the data sets that we have used. The patient data is taken from the Cancer Proteome Atlas, which has 31 different cancer types across varying sample sizes for 12 pathways. And the MD Anderson Cell Lines project is a source from where we have taken the cell lines data, which has more than 650 cell lines and a large set of proteins. We are looking at the 12 pathways, same as the patient data that we are looking at here, and that has 6% missing data, which is a small number, and it does not really have a lot of effect on what we are doing eventually at the end. So we just solve that issue with a simple KNN imputation. This is available across 16 different cell line cancer types. These are the 12 pathways that we have looked at, both functionally and structurally. These are well studied in terms of the cancers that we are looking at, and they were picked mostly as proofs of principles from previous biological studies. For the drug response data, we look at the GDSC or Genomics of Drug Sensitivity in Cancer database. They have 480 plus drugs and across a large set of cell lines, all of which are included in the MCLP. We have looked at a binary version of continuous responses, and the conversion follows the method included in this paper. So this is our framework. Just to remind you once again, this has 3 steps that we are doing. First, fitting the cancer-specific networks and deconvolving to a sample-specific level and getting pathway scores. Second, we do, based on those scores, the cell line identification, and based on those cell lines, we do the drug sensitivity training models. And once we have trained those models, based on that, we can predict the drug sensitivity for the patients. This is a sample result for the hierarchical clustering on the sample-specific pathway scores. In the second row, right here, any dark samples that you see are cell lines and all others are patients. And you can see that there are certain clusters like C4 or C15 or C19 here, which have a sharing between cell lines and patients, based on which you can actually say that these patients are actually similar with these cell lines. In fact, there are certain cancers, for example, the lung cancer and the head and neck cancers, which are actually having their samples separated across 2 different clusters. We actually studied the head and neck samples that were different between the C4 and C15, and we actually saw that they had significantly different survival. So that basically means that our method up to this step is able to identify, even within same organ, same cancer type, different kind of individual cellular signatures that are present in different sorts of patients and samples. Here is the result for the drug response training models that we fit for the cell lines. You can see that clearly across different sorts of cancers, there are different predictive affinities of pathways. Also, here's the result of the predicted drug responses for the patient. What you see in the brackets as the response rates beside each drug is basically the percentage of patients for that cancer type who were predicted to have high response to that drug that we are talking about. And this list is important because all of these drugs that we have listed here that turned out to have high predicted response for the specific cancer types are actually known for those cancer types in literature previously for having successful treatments. For example, ibrutinib is a very standard treatment in breast cancer and has had different sets of clinical trials which have validated its use. So again, we are successfully capturing known signals in terms of drug response to certain cancer types. Finally, we have a large set of resources that we have brought out of this project. This is our published paper. This is our GitHub site where we have all the codes and data which can help you reproduce any of our results presented in the paper. We also have a Shiny app where in the dashboard we basically present all the results that you have just seen and many more, and also the individual networks that we had initially fitted based on the Bayesian graphical regression model. You can also download our sets of codes and a few case studies from here. We also have a YouTube video where we talk about some of the slides that you have seen here, and then in the second half of the video we take you through the Shiny app that you have just seen and basically talk about what tabs contain what information and how best to use that app to understand our results. Finally, a few comments to end. First of all, our method is very flexible in the sense that we can always include more proteins and more pathways because we are doing it at the pathway by pathway basis thanks to larger datasets such as the CPTAC consortium. We can extend to data from other model systems. All the procedure needs to know is that it is patients and some other kind of data. So it does not need to be cell line only. We can also have other omics or transomics data in the regression modeling step. Further, there are some future directions where we can look at. For example, we can theoretically formulate it as a missing data problem, or we can think of this as a labeled unlabeled data problem where the labels are basically the drug responses that we are having that are missing for the patients. These are my references. I'll stop here, and I'd like to listen to and answer any of your questions on this entire deck of slides. Thank you.